In this video, I'm going to talk about the thyroid component of the HPT axis. We are going to be zooming in on the thyroid gland here. Note that this structure is the trachea, so your windpipe, and this is the thyroid gland, which is just to the anterior side. There are ultimately two different lobes connected by this isthmus. So again, if we zoom in, we see that the thyroid gland is actually made up of a series of follicles, and in between those follicles there is various reticular and adipose tissue. There are some parafollicular cells around as well, but in this video we are focusing on the follicles themselves. Inside these spherical structures is a fluid called colloid, and the cells that are making up the thyroid can be called thyrocytes. So if we zoom in on these cells, we can see that on this is the basolateral side, and this is where the colloid would be. Okay, so where the colloid is versus the cells themselves. At all times, the nucleus, or within the nucleus, is occurring the transcription of a gene for something called thyroglobulin. As we know, thyroglobulin is translated in the cytoplasm and ultimately processed by the Golgi apparatus and the RER, and it is released after processing into the colloid. Right? So it's made in these cells and secreted into this fluid called the colloid. Now, thyroid globulin is a fairly long protein molecule, and it has dozens of tyrosine amino acids. Right? So tyrosine residues. Okay. This here is representing a tyrosine, so carbon-carbon nitrogen of the carbon backbone of thyroglobulin, and this here would be the tyrosine R group. Okay. Note that there is a single iodide attached to the tyrosine residue. Okay, so let's go back over here to the follicular cells. Also occurring is the transport of iodide from the blood, from the interstitial fluid, into the cells. This is done by a sodium iodide symporter, the NIS. And so the sodium is continually pumped back out via sodium-potassium pumps, and iodide ends up in the cell. Iodide is then transported into the colloid over here by a protein called pendrin. Okay, so now iodide has been taken out of the blood and is in the colloid over here. Now, once iodine is present, an enzyme called tyrosine peroxidase, TPO, converts iodide into this iodine molecule and completes its oxidation with peroxide. Right, long story short, TPO is then able to attach iodine to the larger thyroglobulin molecule, and so it can start attaching iodines to one of two places. All right, so this here is representing the rest of the thyroglobulin, and here is a particular tyrosine that has been iodinated. Okay, so this particular structure now we can call DIT, diiodotyrosine, so there are two iodines attached. Okay, and so TPO is pretty ambitious with this. It just keeps iodinating anything that it can, and so here we can see it has iodinated these two places on tyrosine to make DIT, and it can also take this entire DIT and join it together with another DIT, so there are two different DIT molecules now, and this entire molecule, again, still attached to the larger thyroglobulin protein. This is called tetra, 4-iodoiodine, or iodine, thyronine. And so tetraiodothyronine is commonly called T4, also called thyroxine. Okay, so that is this and this. Again, it is still bound up to the protein over here, so it's not going to be able to leak out of the follicle and into the blood. Okay. Also, TPO can join together DITs and MITs. Right? So if there's just a single iodine attached to that tyrosine, we call it mono 1-iodotyrosine, MIT. And so on a single thyroglobulin, there might be a series of MITs, a series of DITs, and so TPO is just going to iodinate and join these two molecules together. When there are 3 iodines, as we can see here, this is called triiodothyronine, or T3. In general, this is the more active form of the hormone, but again, it's not being released yet because it is bound up to the rest of the thyroglobulin molecule. In this way, thyroglobulin actually stores these different forms of thyroid hormones in the colloid. So there's this reservoir of thyroid hormones in the colloid of the follicle. Now, where we left off in the last video was talking about TSH. TSH was released from the anterior pituitary, and it's going to bind to TSH receptors on the surface of the follicular cells. TSH is ultimately going to induce a second messenger system cyclic AMP, and this is just a summary, not showing all of the many details. And so TSH is essentially going to increase the synthesis of sodium iodide importers, so this right here, so increasing the amount of iodine that's being taken in. Okay, it's going to increase TPO, this enzyme responsible for iodinating different tyrosine residues on the larger thyroglobulin molecule, and it's going to increase the synthesis of thyroglobulin itself. So up here, transcription, translation, processing, and ultimately loading up that colloid with lots of thyroglobulin. Okay, so TSH essentially increases all of this substrate available to make thyroid hormones. Also, thyroid stimulating hormone is going to induce faster endocytosis. And so what that means here is that all of these thyroglobulins, which are loaded up with T4, with T3, there might be some DIT left, right? There might be some MITs left, okay? And so what's going to happen is that the follicular cells, right, these thyrocytes, are going to endocytose. They are going to endocytose, take in thyroglobulin loaded up with all of these different variants, take it into the cell, okay, and lysosomes, which are circulating throughout the cytoplasm, are going to fuse to this endosome, and they are going to break down the proteins, so proteolysis. And so what they're doing is essentially cleaving the T4 off, liberating the T3, and cutting up the MIT and DIT. Ultimately, the rest of the amino acids, MIT, DIT, and all of the iodide that is associated with these parts of the molecule are going to be recycled. Okay, so all of those tyrosine residues and other amino acids as well as the iodide are going to be reused. They can end up back up here for more protein synthesis, and the iodide can end up out here in the colloid. Now T3 and T4 have been liberated, and they are going to be released ultimately into the blood to continue on to their destination of the target tissues. For the most part, T4 is what is released. About a third to a seventh of the hormone that is released is in its T3 form. Generally, T3 is considered to be more biologically active, but T4 is implicated in a lot of thyroid hormone processes throughout the body at this point. Once T4 gets to a target tissue, it can actually be deiodinated. That is, one of these iodines can be removed, and therefore leaving behind T3. Now, depending on which one of these iodines is actually removed, this one will produce T3, and removing this one will produce reverse T3. Now, regular old T3 is a lot more biologically active, but RT3 is also implicated. It also has some kind of a role. So again, this has been a summary of hormonogenesis, so making of thyroid hormones, both T3 and T4, in response to TSH from the anterior pituitary. Thank you.